Good morning, children. Today we will start with the new topic, which is mitochondria. We have already done some functions of mitochondria when we did electron transport chain in case of plants. So at that time, we did little about the structure of mitochondria. As this is a revision lecture, so it means that we have already done this topic before, but as this is revision lecture going on, I'll again teach you about mitochondria in detail. So I'll start with the organelle mitochondria. Yes, anything which is troubling you, if you are not able to hear me or you are not able to see what is written on the board, just let me know. I'll fix accordingly. If there is any problem with any of you, if you are not able to see the board properly or you are not able to hear me, just let me know. Yes, beta. Any problem with anybody? Are you able to hear me properly? Or the board is also visible? Or is it blurred? You can tell me. Okay, Tadarani, Siksha, fine. Rohan, is it clear? It's visible, fine. Okay, fine. So everybody is saying no problem, ma'am. So fine. I'll continue with the lecture now. Okay. Some of the students have joined late. Is there any problem with the connectivity today or some other reason? Because today the, what I say, very sincere students are joining late. Yashika is joining late. Amrita is joining late. What happened? Is there any problem with the connectivity? Or you woke up late? Okay, no problem. So let's start with the mitochondria. Another name of mitochondria is, as you know, what is it known as? It is known as the powerhouse of the cell. It is also known as powerhouse of the cell. First of all, it was discovered by Kolliker, Kolliker. It was discovered by Kolliker in flight. And where was it discovered? In flight muscles of insects. So in the flight muscles of insects, Kolliker, which was a researcher, he discovered it. Flight muscles of insects means the muscles which are required for flight. You know, the insect, they are able to fly. So those flight muscles, in those flight muscles, these mitochondria were found, first of all discovered. As you know, flight muscles means a lot of work has to be done. That means the insect has to fly to lift its body against the gravity. So a lot of work has to be done. And where a lot of work has to be done, a lot of energy is required. That means that particular site should have a number of mitochondria so that a lot of energy can be produced. So these flight muscles are very rich in mitochondria because they have to produce a lot of energy, and that is only possible if the flight muscles will have a number of mitochondria. So these flight muscles are very rich in mitochondria to provide a lot of energy so that the insect can be lifted up and it can fly against the gravity. So it was discovered by Kolliker in flight muscle, and the term mitochondria was given by another researcher, and the name of the researcher is Benda. Benda was the researcher who gave the term mitochondria. Now, this mitochondria is a self-autonomous organelle. Self-autonomous not self, semi-autonomous. It is a semi-autonomous organelle. It is a semi-autonomous organelle. What do we mean by semi-autonomous organelle? That means it is in a way independent. And why this is semi-autonomous? Because it has its own ribosomes and circular DNA. It has its own ribosomes and circular DNA. So it has its own ribosome and it has the circular DNA. Now, ribosomes are involved in protein synthesis. Their function is protein synthesis. So it can synthesize its own protein. And this has genetic information in it. So it can undergo the process of replication, transcription, translation, all these processes which are required, this can undergo. So the most important processes are carried out by mitochondria on its own. That is why it is known as semi-autonomous organelle. The ribosomes which are present in the mitochondria are 70S type. Where else this 70S type ribosomes are present? 70S type ribosomes are also present in prokaryotes. In prokaryotes also 70S ribosomes are present. So these ribosomes, the same type of ribosomes are present in mitochondria, which are 70S type of ribosome. Now we will study about the structure of mitochondria. How does it look like? We have also done it before, but we are doing it again. So this is one circle, and this is the outer membrane, and then we have the inner membrane, which has these infoldings. And these infoldings are known as cristae. So what do we have in this? We have an outer membrane, we have an inner membrane, the inner membrane has these infoldings, and these infoldings are known as cristae. The infoldings are known as cristae. Besides this, we have this is all matrix. This is all matrix. Okay, then we have the special structures in them, which are ribosomes. Then besides this, we have the DNA, which is a circular DNA. So we have this circular DNA. We have these ribosomes. And we have this circular DNA. We have the circular DNA also. So these are the main structures which are present. Besides this, because of the presence of two membranes, the mitochondria is divided into two compartments. This is inner compartment. And this one is outer compartment. Inner compartment and outer compartment. Now, besides this, we have some special structures which are present on the cristae, and these structures, they appear to be like this. I'll just make on one cristae. Similar structures are present on all the cristaes. So these are known as F0, F1 particles. We have already done this, if you remember, in ETS chain. Or these are also known as elementary particles. Or these are also known as oxysomes. So these are known by these names, F0, F1 particles or elementary particles. Okay, okay, Rohan, I'm just doing it. If the lower part is not visible, I'm just doing it. Is it okay, Rohan, now? Rohan, is it okay now? Okay. So these are various particles which I was talking about. These have three names which I have given, and this is how it looks like. The two compartments are there. Now, one important thing about the two membranes, the outer membrane and the inner membrane, that important feature is this membrane is only permeable, and it has less proteins. The membranes are made up of lipids and proteins. So the outer membrane has less proteins and it is permeable. Whereas this inner membrane, it has more protein and it is selectively permeable. Selectively permeable, and it has more protein. The proteins only help in the transport of substance from outside to inside. So it has more proteins. Priya, I cannot read it, what you wrote on in the text. So no problem. Fine. So no problem, Ache. It's okay. So this is what it looks like, and this is what mitochondria looks like. And one thing more I would like to tell you, this space which is present between the two membranes, this space, I'll just mark it like this by an arrow. This space is known as intermembrane space. This is known as intermembrane space. So is it clear, this structure of mitochondria? Is it clear to you all? Any question in this? Anything which you could not understand? Okay, Priya. Others? Yes, beta. Which you could not understand? Fine. Sadar? Yes. Others? Siksha, fine. Rohan, fine. Okay. Fine. So this is the structure of mitochondria, and we have done that. It is also known as powerhouse of the cell. It is a semi-autonomous organelle because it has its own ribosomes. It has its own circular DNA because of the presence of its own ribosome. It can synthesize its own protein. Same as the ribosomes which are present in prokaryotes. At the same time, because of the presence of circular DNA, it carries all the genetic information and it can undergo the process of replication. Christy, Tadarani is asking Christy. See, I haven't marked. Christy are here. These, the infoldings of inner membrane of mitochondria. This is a Christy. I'll mark it again. Cristae. Cristae are infoldings of inner membrane. This is the inner membrane which has infolding. These infoldings of inner membrane of mitochondria are known as cristae, and these cristae, they bear on them the F0, F1 particles or elementary particles or oxysomes. Okay, these are the real site of ATP synthesis, these F0, F1 particles. Now, one more thing which I would like to tell you, is everything clear to you? What is written? Okay, the image is cleared, it's not blurred now. The image is clear, I think it's not blurred. Okay, fine, fine. So here now, what I want to tell you is, what about this matrix? I would like to tell you something about the matrix. What is there in the matrix? This matrix is very important part of mitochondria. What do we have in this matrix? This I would like to tell you. Here. See, whenever you make a diagram, you need to write the caption over here. Because of the less space, I'm not writing here, but you will have to write here mitochondria. Mitochondria. Okay, and most of the labeling needs to be done on this side. The more labeling should be on this side. Okay, now here, what I want to tell you about the matrix. You know, this the ground substance in the mitochondria, the substance which it is filled up of is matrix. This is matrix. This ground substance is matrix. This is matrix. The matrix of mitochondria, this is rich in, or it has, we will say, it contains the respiratory enzyme. That means this is enzyme, this has, or this contains enzymes for cellular respiration. It contains enzyme for cellular respiration. So all the enzymes for cellular respiration are present in the matrix because it carries out the process of respiration. As a result, ATP is produced. So the matrix should be rich in the enzymes for cellular respiration. At the same time, the matrix has one more thing. This is also rich in divalent cations, divalent ions. Divalent ions like magnesium, Mn, manganese, and iron. This is also rich in these substances. Why these divalent ions are required in the matrix? Although the enzymes are present, but why these divalent ions are also required in the matrix? These divalent ions are required in the matrix because in the presence of these divalent ions, only these enzymes get activated. So to activate these enzymes, these divalent ions are required. These divalent ions, they support the enzymes, or they activate the enzymes so that they can carry out the process of cellular respiration. So this is what the divalent cations does, and that is why these are present here. Fine. Now another important thing about this is, I have already told you that the outer membrane has less protein and this is permeable, whereas the inner membrane has more protein, and this is selectively permeable. The term selectively permeable means it selects the ions, the molecules which needs to pass through this. First, it selects the important ones are only allowed to pass through it, and because it is rich in protein, the inner membrane has more protein. So what type of proteins they help in transport? As we have already done when I told you the fluid mosaic model of plasma membrane, I told you that in between there are tunnel proteins also. Tunnel proteins and other intrinsic proteins are also there, like this. Intrinsic proteins are also there, tunnel proteins. These are tunnel proteins, these are intrinsic proteins. Then we have some superficial proteins also. So I told you these tunnel proteins, they help in the passage of the material from outside to inside. So only the selected molecules are allowed to pass through the inner membrane. That is why we say that this membrane is selectively permeable. Now, we come to the ultrastructure of mitochondria, in which I'll show you this structure, the structure showing the elementary particles or the oxysomes. How does it look like? So, for that, I'll make this membrane and this membrane. So the outer membrane I'll make first. This is the outer membrane. This is the outer membrane. I'm giving it some width, and the inner membrane is here. This has infoldings which are known as cristae. These are infoldings of the inner membrane. So these are cristae. This is outer membrane and that is inner membrane. Now, the inner membrane has two faces. You know, this I have drawn this particular area, this area I have drawn. So matrix will be this side. Here we will have matrix. Okay, matrix will be on this side. Like this, there will be matrix. Okay, I have drawn just a small piece of this. This is outer membrane. This is inner membrane. Inner membrane has two faces. This is the inner is known as M phase. M phase of inner membrane. And this one, this is C phase. C phase of inner membrane. So we have two phases of membrane, the outer phase and the C phase, and M phase, which is the matrix phase, C phase, which is the cytosol phase of inner membrane. So this is what we have in the inner membrane. Now, as I told you, that this inner membrane is rich in proteins and it is selectively permeable. So this membrane has special proteins. Special proteins are present here. And because of this, if you remember, in respiration of plants, we did one ETC. This we did. What was this? If you remember, can you tell me the full form of this? ETC, what was it? Yes, beta, what was ETC? Anybody who can answer? In respiration in plants, we did this. If you remember, what is the full form of ETC? Anybody who remembers ETC? Yes, bache. Electron transport chain or electron transport system. Yes, Tadarani, you are absolutely right. So this is electron transport chain or electron transport system. If you remember, in that we did full complex. Yes, electron transport chain, Yashika, it's right. In this, we did that. There are some, complexes which are present on the inner membrane. We did ubiquinone and many more, CA, C3, C4 complex, CA, all complexes we did. So those, by the help of those, what happens from the matrix, a pair of protons, it moves in. If you remember FADH2, NADH2, and pair of protons was moving here. Pair of protons was moving here. Pair of protons moved here. Pair of protons moved here. So this way, this area, the intermembrane space, it has high concentration of protons, if you remember the electron transport chain. Because of this, the concentration of protons in the intermembrane space, one membrane this, another membrane this, in between is intermembrane space, the concentration of protons was high. Now, as I told you, the cristae has one very important part, which is known as these F1, F0, F1 particles. And these F0, F1 particles, they only help in ATP synthesis. So how they look like. This F0, F1 particle, they have a base. This is the base of that particle. This is the base. Then we have a small elongation of this and a big elongation, big oval structure. Okay, so we have this, the oxysomes, these structures which are looking here bead like, actually they appear like this. And in this structure, this is base. I'm just erasing this matrix which I have written. This is base or F0 particle. Base or F0 particle. This is stalk or F5, F6 particle. This is stalk or F5, F6 particle. And the third one, which is circular and which is bigger in size, this is head or F1 particle. This is head or F1 particle. So this is how the oxysome it appears like. And this F1, this F1 particle, which is also the head, this has the enzyme ATP synthetase. This contains the enzyme. It contains the enzyme ATP synthetase. ATP synthetase is the enzyme which is present in the head part of the oxysome. Now what happens? See, as I told you, the concentration of protons increase in the intermembrane space. One membrane, another membrane. In between intermembrane space, the concentration of protons increase in intermembrane space. So always from the higher concentration, they tend to move towards the lower concentration, and they cannot move through any part, any pore. It's not that these proton can pass through anywhere on this crystal or on this inner membrane. They can pass through from any side. It's not that. These protons, they can only pass through the oxysomes. They can only cross this barrier through oxysomes. So what happens? These high energy protons, they pass through it. When they pass through it, what happens? The energy is released because they are moving from the higher uh, concentration to the lower concentration. The energy is released, and that energy is utilized to combine ADP and P, organic phosphorus. This is combined, and as a result of this combination, the energy is utilized for this. As a result of this, what is synthesized is, as a result of this, ATP is synthesized. ATP synthesis takes place. So this results in the synthesis of ATP. This is how it happens that the ATP is synthesized by utilizing the ATP synthetase enzyme, which is present here. This we did in ETC, electron transport chain. This ETC increases the protons, number of protons in the intermembrane space, and those number of protons, and they tend to move. The number of protons decrease here because most of them, they move here, and this becomes more in the protons. So what happens when they move? They tend to move from the higher concentration to the lower concentration. When they move from the higher concentration to the lower concentration, they can only move through the oxysomes. So the energy is released because they are moving along the concentration gradient. Because of this movement of energy, that energy which is released is utilized in combining ATP with inorganic phosphorus in the presence of enzyme ATP synthetase, which is present in the head region of oxysome, with the release of ATP. So the ATP synthesis takes place, and this is the main thing which happens. This is the one thing which is very important for main function of mitochondria is this, that is synthesis of ATP, because ATP is the energy currency, and this is the only way all the organisms they utilize energy, because ATP is the energy currency in living organisms. Now, this anything in this beta which you could not understand? Anything in this which you could not understand in both the diagrams or here? Yes, you can tell me. Anything which you could not understand in this? Or any query in this? Yes, bache? Okay, Rohan is saying it's clear. To others also. Tadar, fine. Yes. Yashika, Amrita, Priya, Harsha, Siksha. Yes, Siksha, I've got your message. Yes. Anything else? Anybody else? Yes. Deepika. Yes, bache. Jasleen, Tadar. Today the students are less. Yes, bache. Okay, Jasleen, fine. Deepika is not there today. Fine. So this is clear because we have already done ETC when we were doing respiration in plants. So this is based on that. The main is the increase in the number of protons in the intermembrane space. Okay, Harsha, fine, I've got the information. So this is how the ATP synthesis takes place, which is the main function of mitochondria. Now, besides this, there are a few more functions of mitochondria, which I'm going to discuss just now. It is just 3-4 functions are left. So I'll write it here one by one. The first function of mitochondria. The first is ATP synthesis, as we have already discussed it.